about Ebola virus. It's certainly been in the news quite a bit. A lot of people seem to be quite concerned about it. On one hand, there are lots of good reasons to be concerned about it. On the other hand, panic is not a good thing, and probably unwarranted here. So before I get into the details of the Ebola virus too deeply, I just wanted to touch briefly on what a virus is, for those of you who haven't had much experience with them. Viruses are not considered alive. So they are called infectious particles because they don't have the ability to reproduce on their own. Normal cells have organelles, they can make ATP and do all the things that living things do. Viruses can't do that. They absolutely rely on a host cell for making energy, for providing it amino acids for protein synthesis, for providing it nucleotides to make more of its nucleic acid. So they really aren't considered alive. They're really basic in structure for the most part. They have a capsid, so the structure here that most viruses either fall into the icosahedral group or the helical group. The capsid is just a protein structure that encloses nucleic acid. Some viruses, in addition to the capsid and the nucleic acid, have an envelope, a lipid bilayer, that they take from the host cell. And they usually take this as they're leaving the host cell. So that the membrane itself belongs to the host cell. The proteins that stick out from the surface here, those are encoded for by the virus. And it's the proteins that are sticking out that viruses use for attachment to the host cell that they infect. A general replication cycle includes a virus simply attaching to the host cell, and the virus needs access to the host first, but then attaches to the host cell, has to enter the host cell after it attaches, has to lose its protein coat so that the nucleic acid can be exposed to be copied. Then it needs to copy those nucleic acids, needs to make viral proteins or have the cell make viral proteins. Those need to get assembled properly, and then the virus can leave. Depending on the virus, this whole process may take several hours or it can take 20 minutes. It just depends on the virus. And I apologize for not having a really good Ebola replication cycle. So I'm going to use something that is somewhat similar, which is the dengue virus. Back that up just a minute. So the yellow and blue and green things, or not green, yellow, blue, and red things are those are envelope proteins that stick out from the surface of the viral envelope. Interior to that is the lipid bilayer that is from the host. Interior to that is the protein capsid. Interior to that is the viral nucleic acid. So all of those things are important to the virus. So once it gets exposure to the host, it will bind to the surface of the host cell. The attachment proteins on the virus will bind to a very specific receptor on the surface of the host cell. So it doesn't just bind anywhere. Once it binds those receptors, that will trigger the host cell to bring that particle inside. This is a normal cell process that cells use to bring stuff inside. Once it's inside, it's in what we call an endocytic vesicle. In that endocytic vesicle, a few things will happen. Many times a cell will fuse a lysosome with the endocytic vesicle. It'll drop the pH of that vesicle, which will cause some conformational changes in the surface proteins of the virus. When that happens, some hydrophobic portions of the envelope proteins of the virus will fuse with the endosomal membrane and actually kick the capsid then into the cytoplasm. A A variety of things can happen to cause virus uncoding, which is the loss of the capsid, and exposure of the nucleic acid to the cytoplasm. The nucleic acid will then either be copied by a viral enzyme and then expressed by a cell as mRNA, or it might be expressed directly by the cell as RNA if it's something called a positive sense virus. Some viruses, their proteins are expressed as one long polyprotein, which is then chopped up by a protease, a viral protease, autocleaves and then chops up that protein. One of the other things that the virus will make, especially if it's an RNA virus like the Ebola virus, they make something called an RNA-dependent RNA polymerase. That RNA-dependent RNA polymerase is the enzyme that is required to copy an RNA genome. And here it is copied. So it will make a complementary copy of the viral genome, and then it'll use that complementary copy as a template to make more of the original viral genome, shown here in green. So the length of time that takes will vary depending on the size of a viral genome. Those can range anywhere from a few thousand bases to a couple million bases. The capsid proteins will assemble at this point on the edge of the endoplasmic reticulum. The viral genome associates with the capsid proteins. Then that is pulled into the endoplasmic reticulum, where those surface proteins on the capsid are modified. A protein called a matrix protein, which will bind the capsid to the envelope, are added. And that'll move through the Golgi, and then the virus can be released outside the host cell. The number of viruses that can be released from a host cell can be a couple hundred to 100,000 per host cell. So for the specifics of the Ebola virus, the Ebola virus is one of 3 genera in the family Filoviridae. The other 2 members are Marburg virus and Cueva virus. There are 5 species of Ebola virus: Sudan, Reston, Bundabujo, Thai forest, and the one that's the big one right now is one called the Zaire Ebola virus. The virus that's causing problems right now in West Africa is shown here in red. It is a Zaire Ebola virus, but it does form its own clade, so it's a different strain than we've actually seen before for this virus. So the Ebola virus is what we call a negative sense RNA virus. Its genome is a single strand of RNA that's about 19,000 bases long. It's called a negative sense RNA because if a cell tried to translate it as a piece of RNA, it wouldn't make any sense. It's not the coding strand for a message. A complementary strand has to be made in order for this virus to replicate. So when we look at the capsid structure, or the viral structure of this virus, we have the nucleic acid, which is inside this green stuff. The green stuff is the capsid. It's a protein that binds to the nucleic acid. It's referred to as the nucleoprotein, coded for by that gene there. External to the capsid, we have these brown dots, which are matrix proteins, which associate the capsid with the viral envelope. A minor capsid protein, VP24, also facilitates that. The viral polymerase, so the enzyme that actually copies the genome, is packaged in the capsid with this virus. It requires a little subunit, the polymerase cofactor VP35 that's listed there. Embedded in the envelope is this structure here. GP stands for glycoprotein. I'll talk about the SGP, which stands for soluble glycoprotein, in just a minute. The cell types that are infected by the Ebola virus are quite varied. So it can infect a couple types of white blood cells, macrophage, dendritic cells, and T lymphocytes. But it can also infect mucosal epithelium, lymph node epithelium, hepatocytes, fibroblasts, and endothelial cells. So there are many cell types that this virus can infect. The reason why it can infect so many is it actually uses a couple different receptors to get into the host cell. So that glycoprotein on the surface of the virus can bind to one of 3 different receptors that it can use to get into a cell. So that makes it fairly successful in infecting several different cell types. When the virus replicates, it uses its viral proteins on the surface, that glycoprotein, to attach to the receptor on one of those host cells. The receptors that it uses, one's called TIM1, and then there are DC sign, that's hygiene, refers to a receptor on dendritic cells. There are similar molecules with that SIN designate or SIGN designation on hepatocytes, on fibroblasts, and on lymphocytes. So there are a variety of cells that express those receptors. It will bind to those receptors, and then depending on which receptor type it's bound, it can enter the cell via something called receptor-mediated endocytosis, which is one of the ways that cells normally bring things in, or a process called macropinocytosis. That is how cells bring in larger molecules and proteins and things like that. Once it's inside the cell, it is in an endocytic vesicle. There are a couple proteins that the host produces, the cathepsins, that are on the interior of the endosome. Those will interact with the receptors on the those will interact with the glycoproteins on the surface of the virus, causing some conformational change. A lysosome fusion with that endosome will drop the pH, causing some more conformational change, which allows the viral envelope, shown in blue there, to fuse with the endosomal membrane. So rather than the thing inside the endosome being digested, it's actually ejecting the capsid and the viral nucleic acid into the cytoplasm of the host cell. Once that's there, the viral polymerase will make a complementary copy of the genome. It will use that as a template to make more of those negative sense strands, and it will also use that as mRNA for translation to make viral proteins. So the transcripts are shown in these different colors here. Those are translated and made into the viral proteins. Some of the viral proteins will be processed in endoplasmic reticulum. Some of them are processed, or continue to be processed in the Golgi. The glycoprotein will then be embedded in the host cell membrane. The matrix proteins are aligned here on the interior of the cell membrane, the host cell membrane. When the genome is assembled with a nucleoprotein, it then travels to the interior of the host cell membrane, interacts with those matrix proteins, and then exits by a process we call budding. So it actually takes part, so the green part, that's the host cell membrane, pulls that out with it, with the viral proteins embedded in it. The other little dots here are the soluble glycoprotein. One of the products of viral protein production for this particular virus is not all of the glycoprotein that's produced, not all the viral protein that's produced, will be embedded in the host cell membrane. Some of it is simply secreted out of the host cell, not attached to anything. This is one of the more important aspects of this viral infection in terms of the pathology of the disease. So we've got the virus replicating. How does that translate into the horrific disease we know as Ebola hemorrhagic fever? As a virus replicates, especially if it's taking host cell membrane with it as it leaves, that does cause some cell damage, and eventually those cells will die. That's actually only a minor part of the disease process with this virus. That soluble glycoprotein, so that little protein that wasn't embedded in the viral envelope, those can actually bind clotting factors and cause lots of little clots to form in blood vessels. So that's the intravascular, the disseminated intravascular coagulation, or DIC. That's simply because these viral proteins are binding up those factors. This is one important process in hemorrhagic fever. If the platelets are all bound up this way, they're not available to use for typical clotting. The other issue with Ebola virus is that it triggers something called a cytokine storm. And this was a graphic from one of the recent journal articles that discussed cytokine storm. So all of these little dots here are representing cytokines. Cytokines are chemicals that are used to tell cells to do things. In the case of an immune response, these cytokines are used primarily to initiate an inflammation response. Every time we are exposed to something foreign, the first thing that happens in your immune system is you develop an inflammation response. So most of you are probably aware of that if you sprain your ankle. Very quickly, your ankle will swell up because of increased vascular permeability. There will be heat in that region, so fever, localized fever anyway. So it'll be hot, it'll be swollen. This is actually a useful thing because it's bringing in white blood cells to try and fix that damage and come in and clean up any pathogens that might be there. The increase in temperature is an attempt to have that higher temperature be outside of what most pathogens enjoy, their body temperature. So by popping the temperature up a little bit, having that inflammation response, that's generally a good thing. Unless it doesn't stop. In the case of Ebola hemorrhagic fever, the really, really nasty strains of flu that kill people, is the cytokine storm that actually does this. It's the immune system that's really damaging these folks. Normally, in an infection, you'll have an inflammation response that precedes what we call adaptive immunity. Adaptive immunity is the development of immunity that allows for production of antibody and immunologic memory. So the ability to respond really quickly to something you've been exposed to before. So a normal inflammation response is followed by adaptive immunity, so production of antibodies. Cytokine storm, you have these inflammation signals kind of stuck in this feedback loop so that the inflammation response is far greater than normal and it doesn't drop off like it should. There are a couple of reasons for that. The cells that are infected, so T lymphocytes and dendritic cells, those are both very involved in triggering adaptive immunity. So they kind of start that cascade, and so if they're damaged or not functioning properly, we don't get a functional adaptive immunity. Also, some of those cells, some subsets of those cells, are responsible for the attenuation of the inflammation response. When you take those out of commission, nothing is telling the inflammation response to stop. So you get this rather overwhelming inflammation response. So when these types of reactions are going on, you have this great inflammation, and this inflammation, rather than being localized to one spot, it's being brought to the infected cells. The infected cells are in the liver. They're associated with the lymph nodes. They're associated with connective tissue, where the fibroblasts are. And they're associated with endothelial cells that line blood vessels. So damaging all of those things leads to a system that is very permeable, has a lot of holes in it. So that causes that vascular leakage. So blood, plasma, those types of things are leaking out of tissues. The platelets are bound up by the soluble glycoprotein, so they can't do their job. And you end up with a patient that's hemorrhaging. So this is where, these are the things that lead to all the damage in a hemorrhagic fever. So somebody that contracts Ebola virus and is infected with Ebola virus, there's a certain incubation period. That incubation period is anywhere between 2 and 21 days. That's dependent usually on how you got infected and how much virus you got infected with. The first symptoms are fairly nondescript. So fever, muscle aches. At that point, occasionally you start to see a little bit of odd bruising. That moves on to a spiking fever, throwing up of blood, development of massive diarrhea, also full of blood, a lot of bruising, brain damage can happen. The descriptions of people bleeding everywhere are not overblown in this case. If the patient's adaptive immunity response doesn't kick in at that point, then the patient dies. So this is why the transmission of this virus, when they say it's the infected body fluids that are able to transmit this virus, this is why. And the patients are putting out a lot of infected material. For healthcare workers, we're usually taught what we call universal precautions. These things were developed to deal with diseases like HIV, hepatitis B, also acquired by contact with infected material. The sheer volume of material that folks are exposed to here is truly staggering, and that's why so many healthcare workers get infected. So healthcare workers, burial crews, folks that are actually burying the bodies, And lab personnel are at exceptionally high risk for contracting this disease when they're taking care of these patients. This is a picture from the journal Science recently, and I kind of cringed when I saw it. Basically, what they're doing is they're actually testing out a new PCR detection system for Ebola virus, trying to get a quick confirmation on whether somebody is infected or not. And you take a look at this. So this is actually in, it's a lab, mobile lab in Guinea. And you can see we've got folks gowned up. They've got these great protective boots that you can hose off with disinfectant. But I'm cringing because although they're gowned up and gloved up, there's no eye protection, there's no skin protection, and this guy is touching his face with his gloves. So it made me cringe, but it's a great way to sort of show, if you are not careful, something as small as that with this virus can cause an infection. So this virus, one of the ways that infected material can get into you is through your conjunctival membranes. So that's really not a great thing to do. So I'm really hoping that in this case they're dealing with just DNA and not other material. Once somebody is infected, in order to try and control further transmission of that disease, and I realize you can't see all the details on this slide, that's not all that important. What you need to do is figure out who's infected and who they've contacted and where they've went. So some of the really early patients, this was a chart that was actually developed to track who was infected, who they contacted, and who died. This is a real typical type of contact tracing that one needs to do with a disease like this. So if somebody is exposed or may have been exposed, you need to know who they are, where they're going, who they're contacting. The reason for that is if you get infected, if you get infected, you are infectious, you can spread virus to others once your symptoms start showing. So at that point, your viral load is high enough to pass it to somebody else. For the folks that recover from Ebola virus, they may shed virus up to 7 weeks. That's the far end of that window, but it's possible to shed virus for that long. So knowing where people are and what they're doing is kind of important. Prevention of Ebola virus. If you're working in an area with Ebola patients, clearly hygiene is important. For most of the folks that are working in these situations now, you can see that they've got these boots that you can walk through disinfectant and spray down all the time. They've got gowns that cover, or scrubs that cover their entire body. Their heads, and they've got a cowl that actually runs down so that you can't get stuff in your collar. Complete head coverage and your chemistry goggles, basically, so that there's no way for anything to get under there and into your eyes. When you're gowning up and getting rid of your gowns, the buddy system is used. So whenever folks are gowning up and getting out of their gowns now, you don't do it alone. You always do it with somebody else so they can help you out getting certain things off so you don't spray yourself with virus, but it also checks and makes sure that there's nothing showing when you gown up. So that's something that's really important and is continuing to be very important. As far as avoiding it in the first place, this can be a little bit tricky. We don't know exactly where this virus comes from. We'll get to that, more of that in just a minute. But we do know what types of animals can be infected. And those are often consumed as a meat source. It's referred to as bushmeat. So primates and bats are referred to as bushmeat. So there, reminding you if you know somebody that's infected, no physical contact. Wherever you're around where it might be, always make sure that you wash your hands, avoid bushmeat. If you do have to eat bushmeat, sometimes you don't have a choice, make sure that it's cooked really, really, really well. And be very careful when you're handling the animal. These types of things will help prevent spread of virus. So in July, so the epidemic was pretty bad already in July. The WHO made a statement, I think it was July 25th, that it would be unethical to use experimental treatments or experimental vaccines on these populations. So, yes, we have an outbreak, but we haven't tested these things. It would be unethical to use them. And in most cases, you'd have a point. But where you have an epidemic where so many people are dying, there was an argument made for compassionate use. Okay, it might be dangerous, but if my chances are trying this and not dying, great. If I'm going to die anyway, I may as well try. Initially, the World Health Organization said, no, we're not going to do this. The Onion, on July 30th, came out with this. They were off by 48. And it wasn't actually the vaccine, it was an experimental treatment. So there's a difference. 2 American healthcare workers were diagnosed with Ebola, brought back to the States, and were treated with this here, ZMapp. ZMapp is composed of antibody to the Ebola virus. So these are antibodies that are actually made from a cell line in the lab, but they are antibodies that were originally shown to be effective in binding Ebola virus and stopping it from getting into host cells. So these 2 folks were treated, and they recovered. 5 more people have been treated with ZMapp. Out of those 7, 5 have recovered, 2 have not. So relatively good odds if you're treated with ZMapp. This is still something that's really untested. It hasn't been put into clinical trials. It's just, wow, let's just try it. This next one is a treatment for Ebola, something called blood therapy. So the people who have survived clearly have antibody that will help them fight the virus. So blood therapy, and there have actually been a couple of people treated this way already, is simply taking blood from people who have survived, recovered, using their sera to treat people who are infected and have an ongoing disease. The hope being that the survivor's antibodies will bind the Ebola virus and stop it from progressing in that patient. A couple other things that are being developed for treatment of Ebola virus. One is a treatment that is called RNAi, or interfering RNA based. Essentially, that's taking a little piece of RNA that's complementary to the genome of Ebola virus, providing that binds to the genome of Ebola virus, creating a double-stranded RNA that the cell will then recognize as being odd and chop up. So basically the genome of the virus gets chopped up by the host cell, and you aren't going to make any more virus at that point. So there's a couple of those being produced. And there's also another approach, a drug that's developed to inhibit the RNA-dependent RNA polymerase that the virus makes. So those are the main avenues of treatment research. In terms of prevention, there are a couple of vaccines that are, actually there's one that they have in safety trials right now and expect to roll that out sometime in December as far as using that in the field. So just give it a shot. Additionally, there are a few more approaches to vaccines. So the VSV based, that actually means vesicular stomatitis virus. And what they do is they take that virus, put the Ebola surface glycoproteins in its envelope, and vaccinate people with that so you can develop antibodies against those surface glycoproteins. VSV creates a brief infection in the person that's received the vaccine, but you get a really good immune response against those proteins. Same thing with these adenovirus-based vaccines. Just a different virus, you get a brief replication and develop immune response against the protein for Ebola. 5 of them are slated for clinical trials in January. So these are moving forward at a record pace. Generally, it takes a much longer period of time to get those to clinical trial. So why is this one so bad? Previous epidemics were bad at the time. So the bad epidemics affecting somewhere between 300 and 400 people. Ebola virus was discovered in 1976, and these are literally all of the outbreaks since then. They all pale in comparison to what we're seeing now. Where does the virus come from? This is one of the problems. We don't really know. There is a lot of evidence that fruit bats are the reservoir for this virus. We know that the virus can infect a variety of animals, so humans, primates, those fruit bats, pigs can be infected, and if there's any, Chris, is that called a doiker or duper? How's that pronounced? Do you know? Okay. So those things. Diker?
It's antelope.
Okay. Yeah, so small little deer-like thing. We know those animals can be infected. The fruit bat is a slightly better option because the fruit bat you can actually infect with these viruses and not produce an infection that kills the bat quickly. They don't show the Ebola-like symptoms that we see in people and some of the primates. So they can have the virus replicating. They don't die quickly, gives the virus a chance to spread, which is what generally parasites want in a host. Not killing it really quickly, long enough for it to spread a lot. So fruit bats make a good possibility for that. The areas where we're seeing these infections, so West Africa is new. We hadn't seen infections in West Africa before. But prior to that, Democratic Republic of the Congo, Sudan, Kenya, Uganda, and Gabon had all shown outbreaks at some point, either with the Zaire Ebola virus, which is shown in green, the Sudan Ebola virus in blue. Reston Ebola virus is actually in the Philippines, which clearly isn't shown on this map. The Bundabujo strain was just seen here, and the forest Ebola virus, which used to be called the Côte d'Ivoire Ebola virus, that's why it's CI here, was just in this high forest region of the Ivory Coast. The three bats that are most likely are the ones shown here. We've got the yellow-collared bat, the African fruit bat, and the hammerhead bat. These are all three capable of maintaining replication of the virus without developing obvious disease. In nature, we've recovered bats that have antibody to the Ebola virus from all three of these. However, we haven't found a single one that's actually had the virus in it. So there isn't definitive proof that this is where the virus is coming from. But the other data, combined with their range, so Democratic Republic of the Congo, moving over to West Africa, same thing for this one. This one doesn't quite make it to Sierra Leone, Liberia, and Guinea, but it comes close. The other thing that's kind of supporting this is that a closely related virus, the Marburg virus, we actually have found one bat to support that. They actually did find Marburg virus in the Egyptian bat, which is shown there at the bottom. Once. So, current epidemic is by far the worst in history. Previous epidemics were about 6 months in length, and the combined total for all of the Ebola epidemics was 2,417 cases and 1,590 deaths. Right now, we're looking at 9,936 cases, 4,877 confirmed deaths. This area was not really well set up to deal with this type of epidemic. One, they weren't expecting it. They hadn't seen Ebola there before. It's a new place for it. Guinea had the fewest hospital beds per capita in the world. Liberia averages 1 doctor per 71,000 people. Sierra Leone already had the lowest life expectancy globally. These are not economically advantaged areas. They were already stretched as far as their ability to deal with average healthcare situations, as well as a lot of the infectious disease that is endemic there. Things like malaria, salmonella, shigella, things like that. So it was set up, basically, all these situations. I hate to use the term perfect storm for that epidemic, but that's what it was. Nobody's talking about it, but I wanted to point it out. There's also an Ebola epidemic going on in the Democratic Republic of the Congo. It's really not getting much press because West Africa is so much worse right now. But there is also a current epidemic in Democratic Republic of the Congo. It's a different strain than we're seeing in West Africa. So graphically, this is what we're looking at. We are on an upward trajectory here for cases and deaths, and it doesn't look like it's going to turn around anytime soon. Worst case scenarios are saying by the time this finishes, we're looking at 1 million cases. The most optimistic now are right around 30,000. So it is a very serious problem in West Africa. So here, are we prepared? On one hand, we could say not really, based on what happened in Texas. So you had somebody from Liberia walked into a hospital with fever, aches, a lot of the early symptoms of Ebola, stated he was from Liberia, and they sent him home with antibiotics. A couple days later, it comes back, oh look, it might be Ebola. The nursing staff there wasn't trained to deal with it. They have typical gowns and masks, but most hospital staff are trained in typical universal precautions. They're not trained for Ebola. The hospitals where that was the case, so we have several hospitals that can take patients, keep them in an isolation that is appropriate, give them care that's appropriate, and have appropriate protection for the healthcare workers. We do have those facilities in the United States. The closest one to us is in Missoula. But we can deal with it. So we've treated at least 8 Ebola patients at this point, and only one of them has passed the disease on to anybody else. So while we made mistakes, we've also been pretty good at it. We certainly have the resources to deal with it. The CDC has updates almost every day on how to better deal with Ebola. Even CWU has a statement about Ebola. So we're following the CDC guidelines for dealing with Ebola. Kind of interesting is when I'm going to scroll down. I know that's kind of obnoxious when you're watching. But the advice is self-monitor for symptoms if you think somebody's had exposure to Ebola. But as far as what we are told to do, we want to separate the patient in a room with its own bathroom and use proper infection prevention and control measures. Standard contact and droplet precautions if Ebola is suspected. They do need to update this page. But right now, if you're a college administrator and you go to the CDC, look for what they're telling colleges to do, this is it. I'm not sure that our student health center is set up to do this, to contain Ebola virus. And that's not a slam on the student health center. That's not really what we want happening. So there's still some issues with information. States in red are those who've actually contacted the CDC about dealing with potential patients. We've had some rather poor responses to the fact that we have Ebola patients. In Strong, Maine, and also in northern Idaho, these folks were at the same meeting, so there was a Common Core education meeting in Dallas. These elementary school teachers were there representing their schools at this meeting. Then they go home, they're told they can't come back to work because they were in Dallas. Yes, the meeting was like 10 or 15 miles from the hospital. Navarro College in Texas is denying admission to anybody, to any student applying from Africa, the continent, not just the affected countries. 3 relatives of one of the nurses who were just cleared at Kent State have been told to stay away. An LSU employee that actually went over there to train Liberian police in how to deal with this, didn't have any contact with Ebola patients, was told not to come back to work for a while. Syracuse University invited a Pulitzer Prize-winning writer who had been in West Africa covering the story, comes back, gets an invitation, and then they rescind it because he had been in West Africa. And in a title worthy of the Onion, this is the Washington Post. So yeah, there are better ways of dealing with this than that. So at this point, there's good news and bad news. Bad news, last night there was a press conference saying that there is a New York doctor who had treated patients in Guinea who has been diagnosed with Ebola. Mali has its first case of Ebola. Mali borders Guinea. There's a lot of collateral damage, not just straight from Ebola, but other healthcare in West Africa is suffering. They're dealing with so much Ebola, they can't do things like childhood vaccinations. Vaccination rates for children were actually really good in Guinea, about 97%, which is really good. It's far better than we are. Since Ebola started, that's dropped down to about 27%. Any other healthcare? Same thing. The areas that are dealing with it just don't have the personnel to deal with these other things. People who want to send doctors have a hard time doing it. Most hospitals want their doctors to go and help with crises. In this case, if you do that, you've got to have your doctor gone for 2 weeks for training, probably 6 weeks in West Africa, and then 21 days after that to make sure that they aren't infected. So you lose your employee for a good 3 months at a minimum if you send them. Most hospitals are not recommending that their doctors help in help with the Ebola crisis. On the other hand, there is some good news. So I mentioned already there are 5 more vaccines trials slated for January. The EU yesterday pledged €1 billion in aid. Paul Allen, 100 million. Gates Foundation, 50 million. There were cases in Nigeria and Senegal. They have contained these. Nigeria did an amazing job. They had somebody come in, 19 people were infected, and they stopped it. Their contact tracing encompassed about like 26,000 people. It was truly, truly impressive. The 2 Texas nurses have been declared Ebola-free. So there's definitely good news. To put the situation there in a little bit of perspective, this data is a little old. This number should be a little bit higher, not that much higher. HIV is still a far greater problem in Africa than Ebola. So is malaria, starvation, tuberculosis, and even syphilis. So Ebola is horrific. We need to get a handle on it, but it is not the only problem that is there. Do we really need to worry about it here? If you are a healthcare worker and you decide to go, that's one thing. Your chance of getting infected here is very, very, very small. Very small. In other words, your chance of hooking up with Kardashian are far greater than getting Ebola.